Chapter 36 Sensory Systems 36.1 Sensory Processes. By the end of this section, you will be able to do the following. Identify the general and special senses in humans. Describe 3 important steps in sensory perception. Explain the concept of just noticeable difference in sensory perception. Figure 36.1. This shark uses its senses of sight, vibration, lateral line system, and smell to hunt, but it also relies on its ability to sense the electric fields of prey, a sense not present in most land animals. Credit. Modification of work by Hermanus Backpackers Hostel, South Africa. Introduction. In more advanced animals, the senses are constantly at work, making the animal aware of stimuli, such as light, or sound, or the presence of a chemical substance in the external environment, and monitoring information about the organism's internal environment. All bilaterally symmetric animals have a sensory system, and the development of any species' sensory system has been driven by natural selection. Thus, sensory systems differ among species according to the demands of their environments. The shark, unlike most fish predators, is electrosensitive. That is, sensitive to electrical fields produced by other animals in its environment. While it is helpful to this underwater predator, electrosensitivity is a sense not found in most land animals. Chapter outline 36.1 Sensory Processes 36.2 Somatosensation 36.3 Taste and Smell 36.4 Hearing and Vestibular Sensation 36.5 Vision. Senses provide information about the body and its environment. Humans have 5 special senses: olfaction, smell, gustation, taste, equilibrium, balance and body position, vision, and hearing. Additionally, we possess general senses, also called somatosensation, which respond to stimuli like temperature, pain, pressure, and vibration. Vestibular sensation, which is an organism's sense of spatial orientation and balance, proprioception, position of bones, joints, and muscles, and the sense of limb position that is used to track kinesthesia, limb movement, are part of somatosensation. Although the sensory systems associated with these senses are very different, all share a common function: to convert a stimulus, such as light, or sound, or the position of the body, into an electrical signal in the nervous system. This process is called sensory transduction. There are 2 broad types of cellular systems that perform sensory transduction. In one, a neuron works with a sensory receptor, a cell, or cell process that is specialized to engage with and detect a specific stimulus. Stimulation of the sensory receptor activates the associated afferent neuron, which carries information about the stimulus to the central nervous system. In the second type of sensory transduction, a sensory nerve ending responds to a stimulus in the internal or external environment. This neuron constitutes the sensory receptor. Free nerve endings can be stimulated by several different stimuli, thus showing little receptor specificity. For example, pain receptors in your gums and teeth may be stimulated by temperature changes, chemical stimulation, or pressure. Reception. The first step in sensation is reception, which is the activation of sensory receptors by stimuli such as mechanical stimuli, being bent or squished, for example, chemicals, or temperature. The receptor can then respond to the stimuli. The region in space in which a given sensory receptor can respond to a stimulus, be it far away or in contact with the body, is that receptor's receptive field. Think for a moment about the differences in receptive fields for the different senses. For the sense of touch, a stimulus must come into contact with the body. For the sense of hearing, a stimulus can be a moderate distance away. Some baleen whale sounds can propagate for many kilometers. For vision, a stimulus can be very far away. For example, the visual system perceives light from stars at enormous distances. Transduction. The most fundamental function of a sensory system is the translation of a sensory signal to an electrical signal in the nervous system. This takes place at the sensory receptor, and the change in electrical potential that is produced is called the receptor potential. How is sensory input, such as pressure on the skin, changed to a receptor potential? In this example, a type of receptor called a mechanoreceptor, as shown in Figure 36.2, possesses specialized membranes that respond to pressure. Disturbance of these dendrites by compressing them or bending them opens gated ion channels in the plasma membrane of the sensory neuron, changing its electrical potential. Recall that in the nervous system, a positive change of a neuron's electrical potential, also called the membrane potential, depolarizes the neuron. Receptor potentials are graded potentials. The magnitude of these graded, receptor, potentials varies with the strength of the stimulus. If the magnitude of depolarization is sufficient, that is, if membrane potential reaches a threshold, the neuron will fire an action potential. In most cases, the correct stimulus impinging on a sensory receptor will drive membrane potential in a positive direction, although for some receptors, such as those in the visual system, this is not always the case. 1018 Chapter 36, Sensory Systems access for free at openstax.org. Figure 36.2, A. Mechanosensitive ion channels are gated ion channels that respond to mechanical deformation of the plasma membrane. A mechanosensitive channel is connected to the plasma membrane and the cytoskeleton by hair-like tethers. When pressure causes the extracellular matrix to move, the channel opens, allowing ions to enter or exit the cell. B. Stereocilia in the human ear are connected to mechanosensitive ion channels. When a sound causes the stereocilia to move, mechanosensitive ion channels transduce the signal to the cochlear nerve. Sensory receptors for different senses are very different from each other, and they are specialized according to the type of stimulus they sense. They have receptor specificity. For example, touch receptors, light receptors, and sound receptors are each activated by different stimuli. Touch receptors are not sensitive to light or sound. They are sensitive only to touch or pressure. However, stimuli may be combined at higher levels in the brain, as happens with olfaction, contributing to our sense of taste. Encoding and transmission of sensory information. 4 aspects of sensory information are encoded by sensory systems: the type of stimulus, the location of the stimulus in the receptive field, the duration of the stimulus, and the relative intensity of the stimulus. Thus, action potentials transmitted over a sensory receptor's afferent axons encode one type of stimulus, and this segregation of the senses is preserved in other sensory circuits. For example, auditory receptors transmit signals over their own dedicated system, and electrical activity in the axons of the auditory receptors will be interpreted by the brain as an auditory stimulus, a sound. The intensity of a stimulus is often encoded in the rate of action potentials produced by the sensory receptor. Thus, an intense stimulus will produce a more rapid train of action potentials, and reducing the stimulus will likewise slow the rate of production of action potentials. A second way in which intensity is encoded is by the number of receptors activated. An intense stimulus might initiate action potentials in a large number of adjacent receptors, while a less intense stimulus might stimulate fewer receptors. Integration of sensory information begins as soon as the information is received in the CNS, and the brain will further process incoming signals. 36.1. Sensory processes. 1019 Perception. Perception is an individual's interpretation of a sensation. Although perception relies on the activation of sensory receptors, perception happens not at the level of the sensory receptor, but at higher levels in the nervous system, in the brain. The brain distinguishes sensory stimuli through a sensory pathway. Action potentials from sensory receptors travel along neurons that are dedicated to a particular stimulus. These neurons are dedicated to that particular stimulus and synapse with particular neurons in the brain or spinal cord. All sensory signals, except those from the olfactory system, are transmitted through the central nervous system and are routed to the thalamus and to the appropriate region of the cortex. Recall that the thalamus is a structure in the forebrain that serves as a clearinghouse and relay station for sensory, as well as motor, signals. When the sensory signal exits the thalamus, it is conducted to the specific area of the cortex, figure 36.3, dedicated to processing that particular sense. How are neural signals interpreted? Interpretation of sensory signals between individuals of the same species is largely similar, owing to the inherited similarity of their nervous systems. However, there are some individual differences. A good example of this is individual tolerances to a painful stimulus, such as dental pain, which certainly differ. Figure 36.3. In humans, with the exception of olfaction, all sensory signals are routed from the A thalamus to B final processing regions in the cortex of the brain. Credit B. Modification of work by Polina Tashina, scientific method connection. Just noticeable difference. It is easy to differentiate between a 1-pound bag of rice and a 2-pound bag of rice. There is a 1-pound difference, and one bag is twice as heavy as the other. However, would it be as easy to differentiate between a 20 and a 21-pound bag? Question. What is the smallest detectable weight difference between a 1-pound bag of rice and a larger bag? What is the smallest detectable difference between a 20-pound bag and a larger bag? In both cases, at what weights are the differences detected? This smallest detectable difference in stimuli is known as the just noticeable difference, JND. Background. Research background literature on JND and on Weber's law. A description of a proposed mathematical relationship between the overall magnitude of the stimulus and the JND. You will be testing JND of different weights of rice in bags. Choose a convenient increment that is to be stepped through while testing. For example, you could choose 10% increments between 1 and 2 pounds, 1.1, 1.2, 1.3, 1.4, and so on, or 20% increments, 1.2, 1.4, 1.6, and 1.8. Hypothesis. Develop a hypothesis about JND in terms of percentage of the whole weight being tested, such as, the JND between the 2 small bags and between the 2 large bags is proportionally the same, or is not proportionally the same. So, for the first hypothesis, if the JND between the 1-pound bag and a larger bag is 0.2 pounds, that is, 20%, 1.0 pound feels the same as 1.1 pounds, but 1.0 pound feels less than 1.2 pounds, then the JND between the 20-pound bag and a larger bag will also be 20%. So, 20 pounds feels the same as 22 pounds or 23 pounds, but 20 pounds feels less than 24 pounds. Test the hypothesis. Enlist 24 participants, and split them into 2 groups of 12. To set up the demonstration, assuming a 10% increment was selected, have the first group be the 1-pound group. As a counterbalancing measure against a 1020 Chapter 36, Sensory Systems access for free at openstax.org. Systematic error, however, 6 of the first group will compare 1 pound to 2 pounds, and step down in weight, 1.0 to 2.0, 1.0 to 1.9, and so on, while the other 6 will step up, 1.0 to 1.1, 1.0 to 1.2, and so on. Apply the same principle to the 20-pound group, 20 to 40, 20 to 38, and so on, and 20 to 22, 20 to 24, and so on. Given the large difference between 20 and 40 pounds, you may wish to use 30 pounds as your larger weight. In any case, use 2 weights that are easily detectable as different. Record the observations. Record the data in a table similar to the table below. For the 1-pound and 20-pound groups, base weights, record a plus sign, plus, for each participant that detects a difference between the base weight and the step weight. Record a minus sign, for each participant that finds no difference. If 1/10 steps were not used, then replace the steps in the step weight columns with the step you are using. Results of JND testing, plus equals difference, equals no difference, step weight 1 pound 20 pound step weight 1.1221 1.224 1.326 1.428 1.530 1.632 1.734 1.836 1.938 2.040 Table 36.1 Analyze the data. Report the results. What step weight did all participants find to be equal with 1 pound base weight? What about the 20-pound group? Draw a conclusion. Did the data support the hypothesis? Are the final weights proportionally the same? If not, why not? Do the findings adhere to Weber's law? Weber's law states that the concept that a just noticeable difference in a stimulus is proportional to the magnitude of the original stimulus.